Great. Well, thanks for the invitation. As Bert says, I'm working in the cardiovascular epidemiology unit in the Department of Public Health and Primary Care at the University of Cambridge in the UK. Before we start, I should give my disclosures, or what I like to call collaborations of interest. So as you'll see, there's good reason why I work with many industrial partners and also many biotech companies as well in our multi-omics research. So you may want to interpret this talk in that context. Just as background, really, so you can get a sense of where I am. I'm actually at home today, but this is our shiny new building that was built during the pandemic. And as Bert knows, it looks pretty much like it does here, although the duck pond's somewhat more frozen at the moment. So we're in a new Heart and Lung Research Institute, which is on the Cambridge Biomedical campus, so where the clinical school is there, as well as currently 2 hospitals, but soon to expand to 4 hospitals, so a major kind of biomedical research center. And the idea here is that all the cardiovascular and lung research in Cambridge is now centralized in this one building, including computational groups like ours, as well as wet lab groups and people from the departments of medicine and hematology. So as Bert says, the unit has a long history, and perhaps people might know our group from some of its earlier work, particularly in trying to understand relationships, observational relationships between risk factors and outcomes, particularly cardiovascular diseases. So, for example, through the work of the Emerging Risk Factors Collaboration, which was put together by our unit and involved prospective cohort studies from around the world. So here's one example looking at lipoprotein(a) and trying to essentially gather all the world's data on that and to try and provide a clearer picture. So, for example, here we were looking at more than 125,000 participants who had lipoprotein(a) assayed in blood samples, and more than 10,000 incident cardiovascular outcomes that have been recorded. So one could start to look at questions like, to what extent is Lp(a) related to risk of cardiovascular events? What types of cardiovascular events? And is it specific to cardiovascular events? So here we see very little evidence of relationship with non-vascular death, but clear positive relationship at higher levels with higher risk of cardiovascular disease. Of course, more recently, as genomics technology has improved and allowed us to go from measuring one thing very well to measuring millions of things, we've joined with that, and we've tried to use genetic data wherever we can. So in one way, relating genetics to disease outcomes. So here's an example, particularly in coronary artery disease, where we've played a leading role. So this is a paper that I co-led with investigators from Boston, Krishna Aragam and colleagues from the Broad in 2022. Part of a global consortium, Cardiogram plus C4D, where we tried to bring together as many of the disease and non-disease studies as we could in coronary disease. So something like 1 million people, 200,000 cases, and one can see hundreds of different genetic loci for coronary disease. And of course, that points us towards potential new biological insights. What I'm particularly interested in is how we marry those two ideas. So how do we bring together genetic data and the molecular data to inform on disease etiology and causality? So taking the lipoprotein(a) example a step further, on the left here you see analyses we did where we related different genetic variants. So in this case, 43 variants in the LPA gene region. And on the x-axis here, you see their effect on Lp(a) levels in the blood. And on the y-axis here, you see the odds ratio for coronary heart disease. So you can see a relatively clear relationship. So the alleles that give people higher levels of lipoprotein(a) confer a greater risk of coronary heart disease. So we can start to assume a causal relationship rather than just an observational relationship. And one can start to try and assess the magnitude of that relationship as well. So here we took participants who'd been genotyped for these variants, and we said, well, let's create a score out of these variants. So people who have the unluckiest genetic hand, if you like, they're predisposed to having very high Lp(a) levels by their DNA. You can see at the top end here have something like a 1.5 odds ratio for coronary heart disease. So we've been very interested in how can we use genetics to go beyond observational associations, infer causality, and start to understand risk factor disease relationships. And in fact, we use this information to help Novartis then design their phase 3 trial of an Lp(a) inhibitor, which I think should read out in 2025. So we'll get a sense then of how accurate our quantitative causal estimates were from that trial. But of course, as well as the genetics kind of revolution that's allowed us to measure many DNA variants at once, there's been a revolution of a different kind as well, and that's in what we might call multi-omics. So really what my group now tries to think about is how can we fill in that gap between DNA and disease outcomes. So this is a slide that illustrates some of the measurements we've made in one of our main cohorts. We use the interval cohort, which is a collection of about 50,000 blood donors from around the UK who all gave their blood samples for us to store and analyze. And over the years, we've done a series of assays looking at different layers of molecular data. And this has all been as the technology has emerged to allow us to measure thousands or even tens of thousands of things at once, but to do it in a way that's now scalable. So one can do this in hundreds, thousands, even tens of thousands of participants. So among the 50,000 blood donors in interval, we've been able to do a collection of different assays, and some of them we're able to do cohort-wide. So for example, at the right-hand end here, the Sysmex hematology analyzer is run on everybody at baseline on the fresh samples we get. The Nightingale Health Metabolic Panel of about 250 lipids and lipoproteins is relatively cheap and high throughput and affordable, so we can do that in everybody. Whereas, for example, some of the newer proteomic assays like the Olink and SomaLogic technologies that can measure thousands of plasma proteins from a single blood sample, but are much more expensive, and so are in a few thousand people. And over the next few minutes and slides, I want to just illustrate some of the uses of having these data, the kinds of questions that we can try to answer, to give you some examples. But rather than just tell you how wonderful these things are as well, as you may have gathered from the title of the talk, I'd also like to talk about some of the challenges of going from one analyte one risk factor that you measure well and understand and can interrogate in detail, to measuring thousands of things at a time, and the sort of challenges that might bring. So what we can do when we take a platform, a new assay platform, for example, this was the SomaLogic proteomic assay platform that at the time when we first ran this enabled us to measure about 3,500 proteins at a time, and we could do that in about 3,000 participants from the interval study. And one can then say, well, let's take the genomic backbone that we have in interval, so having assayed all of the genetic variation we can, let's try and test that for association against each of the proteins we measure. And this is an attempt to kind of represent that visually. So across the bottom here on the x-axis, we're looking at the position of all the genetic variants that we tested in the genome from chromosome 1 through to 22. And then up the vertical axis here, we're looking at where is the gene that encodes the protein that we were measuring. So each dot here represents a statistically significant association. There are about 2,000 of these that we found overall, even though we only had about 3,000 people. And the red ones here on this diagonal line are what we call cis associations. So that's where, as you might expect, you find a genetic signal like the Lp(a) example I just gave you. You find a genetic signal that's right near the gene that codes for the protein. And one can imagine straightforward ways in which that might happen. So it may, for example, be a genetic variant that alters your gene expression, and that translates into a different protein level in the blood. The bluey, turquoisey ones are what we call trans signals. So that's where you find, perhaps they're more biologically interesting, perhaps. You're finding a signal somewhere else in the genome that relates to the protein level. And you can see there are some kind of vertical bars here. So there are some parts of the genome that are wildly pleiotropic. So for example, on chromosome 9 here, we have the ABO blood group. That seems to be a determinant of the levels of many different proteins. You'll see, perhaps you'll recognize maybe the ApoE locus, for example, a well-known lipid regulator, again, that seems to have effects on many proteins. But many of these individual trans association dots are still to be explained, and to understand, why does a genetic variant somewhere else in the genome affect the levels of the protein we're interested in. So huge potential for biological understanding there. Of course, as I showed you with lipoprotein(a), we were particularly interested in understanding causal relationships. So one approach to do that was to take what had been done already in terms of association and prediction. So here we have a work from 2016 from other colleagues who'd looked at if you took this same proteomic platform and you tried to test it against incident cardiovascular events, either in initially healthy people or in people with stable coronary artery disease, which proteins kind of dropped out from that panel and seemed to be most predictive. And so they created this 9-protein model, which, as you see from the plot, seems to have some ability to differentiate risk here. And in fact, the SomaLogic have been using that in some clinics in the US, I believe. One of the proteins they found was this macrophage metalloelastase, or known as MMP12. And that contributed in a positive way, i.e. higher levels of MMP12 were associated with increased risk of future cardiovascular events. And we thought this would be an interesting one to look at because when we did our genetic association, our genome-wide association study of all these proteins, we found a very strong signal, and we found a strong signal in the region near the MMP12 gene. So you see here at the bottom, this is the position on the genome of the MMP12 gene. And each of these dots represents a genetic variant and its strength of statistical association on the vertical axis here. So you can see some very, very tiny p-values, even though we only have a few thousand participants. So of course, one, as we did with the lipoprotein(a) example, one can then ask the question, well, do people who inherit alleles that predispose them to higher levels of MMP12, do they have higher levels of cardiovascular risk, as one might predict from the observational associations? Well, interestingly, the relationship seems to be in the opposite direction to what we might have hypothesized. So you see from this plot that when we took several independently acting variants, that as the MMP12 levels get higher, the risk of coronary heart disease gets lower. So there seems to be a potentially inverse causal relationship. And this led to hypotheses that actually MMP12 is potentially a protective biomarker here. It's produced by macrophages in response to vascular damage. So perhaps the greater the subclinical atherosclerosis, the more is produced, and therefore the higher levels predict future cardiovascular events. They're in people who are probably greater clinical risk, but actually it's probably, in terms of the activity of MMP12, we think it's probably a protective factor. And this has now been replicated in several different studies with several different protein platforms and several different outcomes as well. So you see similar patterns with ischemic stroke outcomes, for example. So there seems to be a body of evidence that this might be the case. Now that was one what we took. So we'd measured many thousands of proteins, but we were cherry picking one example from our genetic study on the basis of a previous observational association. But of course, the advantage of measuring many different molecular risk factors at once is that one can take a very sort of agnostic and data-driven approach. And that's what we did when the COVID pandemic hit, and we all turned our attention to how could our science try and shed some light on this. So, of course, there were great international efforts to try and do genetic analyses of COVID-19, and an international consortium sprang up very quickly and produced GWAS results for severe disease versus healthy controls or moderate disease. And we could benefit from the international release of those, and we could take our approach of trying to find genetic variants that predict protein or gene expression levels, and try to do if you like, proteome-wide or transcriptome-wide analysis, looking at as many proteins and as many genes as we could. And these are the results we found at the time with the sample sizes we had. So there are a handful of proteins or genes that lit up quite nicely here. Some of these gave us a good sense we were getting the right kind of results. So, for example, the ACE2 protein lit up strongly, which was reassuring because, of course, by this point, ACE2 had been recognized as one of the key cell surface receptors that was crucial for the SARS-CoV-2 virus to infiltrate the cells. But we also found some things that were maybe less well understood from the previous biology. So, for example, we found a signal at IFNAR2, an interferon receptor, which supported some of the literature that was appearing at that time, highlighting the importance of some of the interferon signaling pathways in response to COVID. So we can go from testing kind of one at a time to identify to testing many thousands, and a bit like the GWAS kind of era, taking a very discovery-based approach to see which ones came up. Also, maybe to note here, something I'll come back to, an important additional step, as well as this so-called kind of Mendelian randomization causal inference approach, is to test so-called co-localization. So once one tests many things, we have to be kind of cautious about whether we're really hitting the right target, or whether we might be actually discovering a signal for a neighboring gene or protein. And I'll come back to that in later slides. We've progressed that now with several different diseases. So we're looking at gestational hypertension, for example, glaucoma. One can take these approaches across many different outcomes. But of course, as working in a cardiovascular unit, we're now focusing particularly on cardiovascular outcomes. So this is now trying to do the same kind of thing with coronary artery disease, using the GWAS summary statistics I showed you earlier from our 2022 paper. And then you can see that as with much greater sample sizes, much greater discovery power for the outcome, one can start to find many signals. In fact, we almost have the problem now that when you test 15,000 risk factors, of course, there are many hundreds of significant associations. And of course, the challenge becomes then how to decipher those and how to work out what's real and novel and interesting. So one approach to do that is to identify which regions do we only have a single gene. So you see here there were 86 where we only prioritized a single gene in the locus, and those were ones that hadn't yet been discovered by previous GWAS or MR studies. So we're not just entirely recreating here what's been done using genetics only approaches, but identifying additional signal perhaps. One example I show here is the secretory phospholipase A2 example. So on the left, you see a nice gene expression signal near a cis signal, so near the gene PLA2G5. And on the right, you see the same signal, albeit a weaker magnitude with coronary artery disease. And so one can test these to see in the causal inference framework to see a positive causal relationship, and one can also test for so-called co-localization. So we can perform a statistical test to say what's the probability, if you like, that these signals are actually shared. They're driven by the same underlying causal variant. We might not know exactly which variant it is because there are many quite correlated variants here, but we're pretty confident these are the same signal. And this can, of course, be useful. This perhaps suggests that PLA2G5 might be a good therapeutic target, but it can also help us perhaps understand trial results. So, for example, for Veresplodib in the VISTA trial, which was unfortunately unsuccessful in preventing cardiovascular events, but seems to be a pan inhibitor of these enzymes. So potentially a specific inhibitor of PLA2G5 may have more success than an inhibitor that downregulates many different secretory phospholipases. Now, many of the slides I'll show you are based on the protein and the proteomics, partly because I think that's a very relevant layer to lots of diseases, partly because we've run many different assays on the proteomics, partly because it's one of the ones we looked at first. But I'd also like to show you some examples from other domains as well, because I don't think it's only the proteins and the gene expression layers that are of great interest here. So this was a project we did that run using an untargeted mass spec-based metabolomics platform from the company Metabolon. It's actually 4 kind of slightly separate mass spec assays that they combine into one package. So this gives you about 1,000 different metabolites that can be assayed in a plasma or serum sample. And you may be able to see in the middle of this plot some of the kinds of things that are measured here. So amino acids, carbohydrates, lipids, etc. About 2/3 of those are kind of known and named and labeled, and about 1/3 of those are still remain to be matched to libraries and interrogated or understood, but can still be analyzed in epidemiological studies. So again, we took the same kind of paradigm as earlier. We can relate our genome-wide association data to each of these 1,000 metabolites. And this time we're kind of trying to represent it in a circular form here. So each of the kind of colored rows, if you like, around the circle represents a particular metabolite. And then we're going around the genome here from chromosome 1 to 22. So you can see again some highly pleiotropic loci, some well-known lipid regulators, for example, that show up across swathes of these different tracts. But again, there are many quite specific examples where there are just single dots, perhaps genetic associations that lie near an enzyme that codes for a particular reaction, for example, or a gene that codes for a particularly important protein to a single metabolic process. And so again, by doing this in thousands of people, one can find thousands of independent associations and hopefully not only shed light on biological processes, but disease etiology. So an example here from a different disease area, and this was signals that we found for metabolites, particularly steroids, hormones in the SRD5A2 region. This is a steroid reductase, one of a family of reductases that act on the steroid pathways. And it was interesting because inhibitors of this this reductase are actually used to treat male pattern baldness and other conditions related to baldness. And so it was nice that we saw on the bottom four here, you see a representation of this region around this gene, signals for several different steroid metabolites. We also found a strong genetic signal for male pattern baldness in the region, which was nice and seemed to confirm the what's already known from the effectiveness of these inhibitors in clinical trials. But what was particularly interesting, and we tried to look at, was this signal at the top for depression. And the reason for that is that on the label for these inhibitors, there is an FDA warning that there may be risk of depressive thoughts, and there has been postulated links between use of these inhibitors and suicide. And so we tried to answer the question of, can we tell anything by combining the genetics and the metabolite data with genetic data for disease outcomes or phenotypes? Can we understand about whether this might be a causal effect or not? And so we could look at how these genetic alleles seem to affect the different steroids, the different metabolites we had, and start to understand what are these genetic variants doing in terms of their effect on reductase activity. And then we could put that into the context then in terms of male pattern baldness and depression. And what we saw is that actually the consistent with the baldness direction of the baldness, or sorry, opposite to the direction of the baldness signal, which goes in the same direction as the effect on the reductase, there does actually seem to be this perhaps small increase in risk of depression. And this suggested to us that perhaps the warning, the side effects that have perhaps been seen might be on-target effects here. So there may be some causal link between inhibiting SRD5A2 efficacy for treating baldness, but perhaps an adverse effect on depressive symptoms. So I've told you about a couple of the different panels here individually. Of course, we often use this term multi-omics, where we think about different molecular layers, and ideally one might like to think about using multiple layers, or if you like, trying to, as I said, fill in the gap between DNA and disease. So how can we understand why does a particular genetic variant confer increased or reduced risk of disease? What are the intermediate molecular steps? And so we have made some efforts in that regard as well, rather than just domain by domain. So for example, here's a region of the genome that's associated with coronary artery disease, with several genes in the region, and it doesn't have a necessarily obvious relation to, none of them had a particularly obvious relationship to atherosclerosis and cardiovascular disease. So we hypothesized we might be able to use molecular data to understand the causal chain of events here. So that was partly suggested by the fact that when we looked in our somologic protein data, we could see in exactly the same place a nice plasma protein signal, and that is for this levels of this protein SWAP70. So we have a nice cis signal here that seems to co-localize nicely, so perhaps suggesting this might be the causal gene in the region. SWAP70 seem to be involved in B-cell switching phenotype, which again might then point us towards some kind of biological mechanism. Interestingly, there's also a blood cis eQTL here. So as I alluded to earlier, perhaps for some of these protein signals, we might be able to understand how these changes in blood protein levels occur. So here it seems like it may be related to differential gene expression in the blood, which then leads to altered protein levels. So one can start to go from DNA variant to gene expression to protein levels. Of course, when one looks at when one can interrogate essentially every gene and its gene expression, it becomes important not only to think about the single gene you're interested in, but can we rule out effects of other genes? So this is a plot that tries to look at all of the other genes that one could assay in the region and say, well, which of these seem to be the same signal? So you see the SWAP70 up here at the top. But actually, it didn't look like any of these variants, these brightly colored variants, seem to have strong effects on any of the other neighboring genes. So again, this perhaps gives us slightly more evidence that SWAP70 is the key causal gene here. When one looks across other domains, as well as gene expression and protein, so for example here, hematology traits, we found signals with red cell traits and also with platelet traits. And in fact, then when one looks at cardiovascular risk factors, we can see here there are perhaps multiple signals in the region for systolic blood pressure, but one of them, not this one on the right with the dark blue variants, but again, we can see perhaps an independent signal here with the same lead variant as we've seen for the SWAP70 protein and gene. And so one can start to put this kind of chain of events perhaps into context and direction. And so we showed that the T allele for this particular variant is associated with increased gene expression and plasma protein levels, effects on these hematological traits, reduced blood pressure, and lower risk of coronary disease. So one can then build a kind of hypothesis about a mechanistic pathway. But of course, then one needs to test that. So working with collaborators in our new institute, we're now doing a series of functional experiments, so cellular experiments, mouse models, but also perhaps the bit we're most interested in is recalling individuals on the basis of their genotype to be able to do more in-depth profiling of people than we can just from taking a static blood sample at baseline. So calling some of these blood donors back and collecting new measurements on them as well. So we have some emerging mouse data that suggests we can reproduce the blood pressure phenotype, but hopefully in the coming weeks and months, we'll be able to firm up some of those results and to be able to confirm or refute the hypothesis that's suggested from our genetic epidemiology. So I hope so far I've given you some sense of the value of doing these molecular assays at scale and the kinds of questions we can answer. But as I said at the start, these also come with challenges. When we move from one single risk factor to thousands, there are challenges both in the measurement, but there are also challenges in how we explore, how we understand, and how we even analyze these. So in the remaining time, I'd like to just explain some of the challenges or highlight some of those and illustrate some of these points using examples that we've found previously. So the first question we might ask is, how can we be sure we're measuring the right thing? You know, you send your blood sample off to measure thousands of proteins or 1,000 metabolites. You know, how do we know we're measuring the right thing? Well, of course, the genetics is often highly confirmatory. So we might often see a plot like this where there's really no signal throughout most of the genome, but then we see a strong signal in one place. And as you might guess from my earlier plots, many of these are cis signals. So we see a really strong signal right near the gene that codes for the target protein. And this would give us confidence. But that's not always the case. So we thought this was a nice plot of the protein ALPL. However, when we looked, the gene that codes for that protein was over on chromosome 1. And actually, this signal is right near a gene called LTF. When we discussed this with the assay manufacturers, they went away and did some investigation and found that, oh, lo and behold, when you use this particular assay in blood, it seems like it's not binding to ALPL, but it's actually binding preferentially to LTF. So actually what we were really getting was a GWAS of the LTF protein. And it was through the genetic analysis and subsequent interrogation. So although the company may have thought they were measuring one particular analyte, in this case, it was a different analyte. So one thing that we often have to watch out for. We also might then ask the question, well, what form of the analyte are we measuring? So these assays might often be developed in a nice model system in a purified recombinant protein form with not a messy, noisy matrix like human plasma. And so we might not know exactly what we're binding to. We might be hitting the right target, but what form are we measuring? So this was an example with a again from the SomaLogic assay, which had a binder to proteinase 3. So we seemed to pretty confident we were from the genetic data we were measuring proteinase 3, but what form were we measuring? So actually, when we take that and we do a pull down of that, and then we run on a gel, what's actually binding. You can see here on the left there was some weak binding to the molecular weight of free proteinase 3 levels, but there was much stronger binding to the complex proteinase 3 to alpha-1 antitrypsin that it binds to. So here we were detecting proteinase 3, but much of the proteinase 3 is actually bound in a complex, and a small amount was free. So again, depending upon the analyses we're doing with these measures, we might need to be conscious of that. If we think we are measuring the right thing and we know what form we're measuring, the next question might be, well, are we measuring it accurately? And there are various ways we can assess this. Coefficient of variation is a way that's often used, and for different assay platforms and even different measurements on those platforms, we might see quite different levels. So reassuringly, here in the plot, you see most of the proteins we were quantifying had relatively low measures, so they seem to be reasonably reproducible when you measure the same sample again and again. You know, for example, on each plate, you put the same sample, and there isn't too much variability. So at least for many of these, we're probably getting good signal, but of course, for some days, we're probably really getting just sort of assay noise and be unlikely to detect epidemiological associations. There's also the question about whether we're measuring these things with enough sensitivity as well. So there are huge challenges, of course, in trying to measure many thousands of things in the blood, not least the fact that many of these are at very low concentrations. An example of this is interleukin-6, and this is a protein assay that measured interleukin-6, but many of the samples, in our case, 70% of the interval samples were flagged as being below the limit of detection. So we could only really measure levels above about 1.8, 1.9 here. And what we found then is when we just looked at those samples that are deemed detectable and above the limit of detection, when we took a really well-known genetic variant that's supposed to associate with these, we see no effect at all. We've kind of truncated our distribution. But if we go back and we include all the raw values that were deemed as kind of inaccurate and below the limit of detection, so if we kind of complete that distribution, we bring back in those 70% of samples, we can see that we actually now start to see below that line, we start to see this nice dose-response relationship with genotype. We recreate the known genetic epidemiology here. So we've had lots of debates with assay manufacturers over the years about what limits should really be used, and should we include or exclude values that are often deemed to be below the limits. When you're measuring these things at epidemiological scale, rather than perhaps a small recall experiment or a cellular experiment, we often have challenges due to assaying many thousands of samples. And of course, from lab to lab around the world and company to company, ability to do this well will also vary. So this is an example going back to our hematology assays that I mentioned that were done in these 50,000 blood donors as we collected them. And so you can see here on the left, raw measurements. This is one of the measurements of the volume of the red blood cells. And you can see that we were measuring these over a kind of 3 to 4 year span. And of course, over that time span, there are perhaps things that change. So in this case, machines that were calibrated, and you can see quite clearly recalibration jumps. So by collecting these data and measuring again, being careful to have control samples, and we could start to detect where these jumps were, and we can start to think about how we might correct the measurements so that we can reduce one source of technical variability. This is another example. This is a white blood cell count, and this time we're looking at how long it is from actually taking the blood to actually processing the blood. And so we can see, again, these kind of technical factors, these things that vary from sample to sample, can have some reasonable amount of impact on the measures that you get. And of course, one can then, if one collects good data on it, can think about how to correct for that and therefore perhaps improve your ability to detect effects. Sometimes these might explain trivial amounts of the variation, but actually we've shown for some analytes that these can greatly improve your power to detect effects. So this is actually in UK Biobank now rather than the interval blood donors, but this is using the Nightingale Health NMR platform. We spent a lot of time poring over the QC and the technical variability in these data. And what you see here is that for two particular biomarkers, alanine on the left and albumin on the right, when you correct out a lot of this technical variability, you can see much stronger associations on the vertical axis after you've corrected than on the x-axis in the original biomarker concentrations. So again, ability to measure things reliably over time and to try and remove as much of the non-biological variation as possible can have a big difference. There are perhaps some more esoteric things that we worry about as well. So, for example, a lot of the protein measurements that we work with are based on binding assays. What we're really seeing there is a readout of the amount of binding, and that might not always reflect the concentration of the protein. So, for example, we often worry about genetic variation that gives different proteoforms. It changes the coding sequence of the protein and therefore potentially gives you a different so-called epitope. So a different, it alters where the binding takes place. And therefore what we may be seeing is not actually differences in protein concentrations, but difference in binding ability due to genetic variants. And we've had to spend a long time talking to assay providers about this and to often trying to persuade them that this is a problem. As you see from this poster at a conference, actually some of the companies were kind of keen to promote this as a way that their platform can actually detect a genetic variant, albeit at astronomical cost compared to genotyping a SNP. So this is something that we worry a lot about, particularly because we're interested not so much maybe in the prediction, where maybe you're not so fussed about whether you're predicting based on a protein level or a genetic variants, but particularly when we come to making causal inferences. Now, you saw on my slide, I've measured several different, we've used several different platforms for the same kind of molecular domain. You might say, well, why bother with that? Well, there is a lot of overlap between these platforms, which can be useful, but there's also a lot of non-overlap as well. So we think both the overlap is informative for giving different kind of testing the same thing using different methods, but also the non-overlap can be important as well because it allows us to interrogate deeper into the proteome or the metabolome. So that's one of the reasons to do that. And of course, we've done lots of work trying to look at to what extent do these assays tell the same story. So here you're looking at genetic associations, so just the beta estimates each for a variant to protein association. On the vertical axis, we're looking at an assay from a company called Olink, and on the x-axis, the SomaLogic assay. And so quite often there's a reasonable positive correlation here. So quite often they tell the same story, but actually some of the ones that we saw in the SomaLogic assay seem to have virtually no effect at all in Olink. Such as this one, a nice cis-pQTL for the GDF15 protein. You see here we're in the GDF15 region, and a p-value approximating 10 to the minus 100 using the SomaLogic assay. But when you test this same top genetic variant using the Olink assay, it was completely null, p-value 0.5. So one platform telling you there's an extraordinarily strong signal and one telling you there's no signal at all. And in fact, we believe this is highly likely due to the problem I told you about before, with a coding variant altering the binding of the assay, affecting one platform and where that binds to GDF15, but not the other. So hopefully having multiple readouts from different platforms can shed light on these. And if you're interested in understanding some of the reasons for differences between the assays, particularly in proteins, then our collaborators, Mike Pietzner and Claudia Langenberg in Berlin, have done a great paper comparing these and explaining lots of the different reasons and trying to work out how important they all are. It can also be not just the assays themselves, but of course the participants. So this is a paper that's we have coming out soon in Nature, looking at the Nightingale Health metabolic traits. And we asked the question here, how much does fasting status make a difference? So this is the same plot. On the top, you're looking at people who are approximately kind of fasted for several hours, and on the bottom, people who are non-fasted for the same. In this case, it's for glucose, and you can see there are some signals that are shared, of course, but of course there are also some signals that are only seen in certain states. And of course, here we were comparing to UK Biobank data, which is fantastic. It's a single very large cohort that's accessible to the whole world. But of course, if you only have one study and one data point, you don't necessarily see the full picture. You're restricted to the status that they used in their study. Similarly here, this is for serum or plasma. So you've got UK Biobank on the bottom, 120,000 participants, and a meta-analysis we did of about 30 different studies on the top. And you can see quite some difference. This is for phenylalanine. So we see the PAH gene, as we might expect, lighting up nicely in both. But we also see a series of signals that light up very strongly in some studies, but not in UK Biobank. And we suspect that this is largely due to use of serum or plasma. Finally, just to say that, of course, moving from one analyte to thousands causes computational problems as well, and of course, computing and carbon problems. So, for example, for some of our metabolomics work, we estimated we were doing about 130 billion calculations. And of course, more efficient ways to do that are going to become increasingly important as we become more conscious of our environmental footprint for computing. So challenges there too. So I hope I've shown you that these platforms are capable of measuring many things and many thousands of things and many thousands of people, and that there's lots of great uses of those, particularly for my interest around understanding disease causality. But there are many challenges as well, and so real caution, I think, is warranted, and deep interrogation of when you find interesting signals is often required. And I just finished by acknowledging my group here on the left, who did many of the analyses, as well as John Dinesh and Emanuele D'Angelo Antonio, who built many of the studies and generated a lot of the assays or funding for the assays, as well as colleagues here who've led many of the papers that I've just shown you, and of course our main funders from the NHS, Health Data Research UK, and the British Heart Foundation. And if there's time, I'll take any questions. Thank you.